Scientists have finally analyzed Noah's DNA, and the findings are shocking. Hidden within his genetic code are mysteries that challenge everything we know about human origins. The patterns uncovered are strange, unsettling, and unlike anything seen before, hinting at secrets buried for thousands of years. What these discoveries reveal could change the way we understand history, humanity, and the legacy of the world's most famous flood survivor. Biblical and historical context of Noah's origin. The story of Noah is one of the most famous and enduring tales in human history. In the book of Genesis, Noah appears as a righteous man living in a time when the world had grown corrupt and violent. God, seeing the wickedness of humanity, decided to cleanse the earth with a flood. He chose Noah to survive this great catastrophe and tasked him with building a massive ark. Noah obeyed without hesitation, carefully gathering his family, their wives, and representatives of every kind of animal. When the rain fell and the waters rose, the ark became a floating sanctuary carrying the chosen few through the deluge. After the floodwaters receded, Noah's family stepped onto dry land, beginning a new chapter in human history. The genealogical records in Genesis trace the descendants of his 3 sons, showing how they spread across the earth and became the ancestors of many nations. From Shem came the Semitic peoples. From Ham arose the peoples of Africa and parts of the Near East, and from Japheth emerged groups that settled in Europe and beyond. These accounts emphasize the importance of Noah's family as the foundation for post-flood humanity and highlight the continuity of life through his lineage. Noah's story holds profound significance in religious thought. In the Abrahamic faiths, he is a symbol of obedience, faith, and divine mercy. The flood represents both judgment and the possibility of a new beginning. Through Noah, humanity receives a chance to start over, making him a central figure in the moral and spiritual teachings of Judaism, Christianity, and Islam. The covenant God made with Noah after the flood, promising never again to destroy the earth in this way, is a cornerstone of theological reflection. This covenant carries lessons about responsibility, morality, and the enduring relationship between humanity and the divine. Noah's sons are especially important because their lineages are linked to key figures and nations in later biblical history, giving his family a lasting influence on religious and cultural narratives. Despite the biblical clarity of Noah's story, science and history present challenges. Many historians and geologists question the possibility of a global flood covering the entire Earth. There is no definitive geological evidence for such a worldwide catastrophe, and attempts to locate the ark on Mount Ararat or other locations have produced inconclusive results. Some scholars suggest that the flood story may reflect a large but regional event, possibly in Mesopotamia, which was remembered and passed down through oral traditions. Genetics adds another layer of complexity. If all humans descended from a single family, how could modern humanity display such vast genetic diversity? Researchers point out that the bottleneck implied by the Ark story seems difficult to reconcile with the patterns found in human DNA today. Over centuries, historians and explorers have searched for tangible evidence of Noah's Ark, driven by curiosity and faith, but definitive proof remains elusive, leaving the story suspended between myth, faith, and history. Search of Noah's Ark. Some of the earliest recorded searches came from scholars and travelers drawn by local legends. Mount Ararat in modern-day Turkey became the focal point of attention because of its high peaks and persistent snow cover, which seemed to protect possible remnants of the Ark. Early explorers claimed to see dark shapes among the ridges or fragments of wood embedded in ice. These claims were often difficult to verify, but they fueled curiosity for generations to come. Local communities also passed down stories of large structures or unusual formations on the mountains, giving hope to those seeking physical confirmation of the biblical account. In the 20th century, the search intensified with modern technology and more organized expeditions. Some explorers focused on the slopes and summit of Ararat, hoping to locate wooden remains preserved by the cold climate. Others turned to nearby valleys and geological formations, interpreting unusual shapes as the possible remains of Noah's vessel. Certain formations, such as boat-shaped mounds of rock and ice, attracted particular attention. Satellite images, aerial photography, and even radar surveys were employed to detect unusual patterns beneath glaciers, though the results were often inconclusive. Prominent figures in these searches include explorers who devoted years of their lives to the Ark quest. Some were trained historians or archaeologists, while others came from unrelated professions but were motivated by faith and curiosity. Expeditions often combined scientific methods with local testimonies, combining measurements, photographs, and samples from the terrain. Many researchers reported intriguing findings, such as timbers embedded in ice or anomalies in the rock that resembled ship ribs. Yet mainstream archaeologists and geologists have generally remained skeptical, arguing that natural rock formations, erosion, and human misinterpretation could explain most observations. The quest to find Noah's lineage shifted from the mountain to our very genes, beginning a journey that would challenge both science and scripture in ways never imagined. Why scientists even think about Noah's DNA? Idea of Noah's DNA might sound like something out of science fiction, but it has fascinated both religious scholars and certain genetic researchers for decades. The central question is simple, yet profound. If all humans descended from a single family after the Great Flood, what traces of that ancestry could still exist in our genes today? For scientists who approach the Bible literally, especially those following the young Earth creationist model, this question is not merely theoretical. They assume that human history is only about 6,000 years old and that all of humanity comes from the 8 people who survived the flood: Noah, his wife, his 3 sons, and their wives. This framework sets the stage for examining the genetic implications of such a narrow ancestry and its potential fingerprints in the modern human genome. One of the most important concepts in this research is the genetic bottleneck. In simple terms, a bottleneck occurs when a population shrinks to a very small number of individuals and then later expands again. All descendants inherit the genes of those initial survivors, which can create distinctive patterns of genetic diversity. Applied to Noah's story, this means that the entire human population today might carry subtle signals from those first 8 people. Scientists and researchers exploring this idea look at the founder effect, which is how certain traits or DNA sequences become common because the original population was so small. Patterns in Y chromosome DNA, which passes from father to son, and mitochondrial DNA, which passes from mother to child, become particularly important. These patterns can, in theory, reveal traces of humanity's first families. A few researchers and institutions have taken a leading role in studying this idea. Dr. Nathaniel Jameson, a biologist affiliated with Answers in Genesis, has published extensive research on how Y chromosome and mitochondrial DNA could reflect the history of a post-flood population. His work often compares the observed genetic diversity in humans today with what would be expected if all humans descended from Noah's family roughly 6,000 years ago. Alongside Janson, the Institute for Creation Research has promoted this line of study, supporting analyses that attempt to reconcile genetic data with the literal biblical timeline. Their research emphasizes that modern genetics does not necessarily contradict a biblical view if the right population models are applied. The studies are still controversial and widely debated in mainstream science, but they have opened a unique window into the intersection of faith and genetics. By treating Noah's family as a real historical bottleneck, researchers hope to uncover clues hidden in human DNA that could correspond to the biblical narrative. They look at how lineages expand and how rare genetic sequences can persist over millennia. When Janson and his team turned to mitochondrial DNA, they discovered a surprising detail. The findings sparked intense curiosity and further research, hinting that the story of Noah might leave more than a moral or spiritual legacy. It could leave an imprint in the very building blocks of life itself. Mitochondrial DNA evidence according to creationist research. Mitochondrial DNA, or mtDNA, is a special type of genetic material that humans inherit only from their mothers. Unlike most DNA, which comes from both parents, mtDNA passes directly down the maternal line, making it an ideal tool for tracing ancestry over generations. Scientists can use it to construct maternal family trees and observe how populations have spread and diversified over time. For researchers interested in the biblical account of Noah, mtDNA offers a possible way to test whether modern humans might indeed descend from just a few post-flood families. Dr. Nathaniel Johnson and his team undertook one of the most notable studies in this area. By analyzing hundreds of human mtDNA sequences, Johnson mapped the relationships between maternal lineages and noticed something striking. The tree of mtDNA appeared to have 3 central trunks or nodes. In the creationist interpretation, these 3 main branches corresponded neatly to the 3 wives of Noah's sons. According to this view, the descendants of each wife contributed a foundational maternal line to the human population after the flood. The tree's structure seemed to suggest that humanity today might still carry echoes of these original maternal lines in our genetic code. Another key part of Johnson's analysis involves mutation rates and timing. mtDNA accumulates small changes, called mutations, over generations. By calculating the number of differences between sequences and applying a mutation rate of roughly 1 mutation every 6 generations, Johnson argued that the observed diversity in modern human mtDNA fits a timeline consistent with a young Earth flood model. In other words, the length and branching of the mtDNA tree could match what would be expected if all humans descended from Noah's family just a few thousand years ago. This interpretation contrasts sharply with mainstream scientific models, which assume much older timelines and more gradual accumulation of genetic diversity. However, the approach is not without controversy. Critics point out that Johnson's conclusions rely heavily on specific assumptions about mutation rates, the average age of reproduction, and exact genealogical timelines. Even small changes in these assumptions can dramatically alter the conclusions. Mainstream geneticists remain skeptical, arguing that the mtDNA evidence is compatible with many possible scenarios and does not definitively point to a single historical bottleneck, as described in the Bible. Despite these criticisms, creationist researchers continue to explore mtDNA as a possible window into the history of humanity's earliest families, seeking patterns that might align with the biblical narrative. The mtDNA findings open up fascinating questions. If 3 maternal lines can be traced back and potentially linked to the 3 wives of Noah's sons, what about the paternal side? Could the Y chromosome tell a similar story for Shem, Ham, and Japheth? The possibility of uncovering corresponding paternal evidence promises to deepen the mystery and push the investigation of Noah's descendants into uncharted territory. Y chromosome evidence in the creationist model. The Y chromosome offers a unique window into human ancestry because it passes almost unchanged from father to son. Unlike mitochondrial DNA, which traces the maternal line, the Y chromosome carries the genetic signature of the paternal line, making it ideal for studying how men are related across generations. For creationist researchers exploring the story of Noah, the Y chromosome presents an opportunity to investigate whether all modern male lineages might trace back to the 3 sons of Noah, Shem, Ham, and Japheth. Dr. Nathaniel Johnson and his colleague David Holland conducted an influential study using pedigree-based mutation rates. Instead of relying on theoretical estimates, they measured the rate of mutations on the Y chromosome using real family data, observing how DNA changes occur over known generations. Their results suggested that the observed diversity in Y chromosomes could be explained within a relatively short timeline, roughly 4,500 years, which aligns with the biblical post-flood model of human history. This finding challenges the standard scientific assumption that Y lineages have been diverging over tens of thousands of years. Johnson also examined global Y chromosome phylogenies, the branching family trees that map the relationships between all major male lineages. He proposed a recent origin model suggesting that many branches reflect rapid population growth over the last few thousand years, rather than deep evolutionary time. In this view, the diversity seen in Y lineages today could be the result of the descendants of Noah's 3 sons expanding into a global population after the flood. Some creationist researchers have gone even further by introducing the concept of a Noah root for the Y chromosome tree. Instead of assuming that all modern Y lineages trace back to ancient hominins hundreds of thousands of years ago, they suggest that the root of the human Y chromosome tree begins with Noah's own Y chromosome. From this hypothetical starting point, the 3 sons' Y chromosomes gave rise to all the haplogroups found in modern men. This reinterpretation of the Y tree provides a framework in which genetic patterns are directly linked to the biblical narrative, creating a potential genetic echo of Noah's family in every male today. Of course, these ideas face strong criticism from mainstream geneticists. Critics argue that the mutation rates used in these models are higher than those accepted in conventional research, making the timelines appear artificially short. Rerouting the Y chromosome tree based on a biblical framework is considered scientifically controversial because it assumes conclusions from Scripture rather than letting the genetic evidence speak independently. Many argue that while the patterns of Y DNA are real, their interpretation in terms of a single historical family is speculative. Still, for creationist scientists, the Y chromosome evidence is tantalizing. When combined with the mtDNA patterns, which also hint at 3 maternal lineages, the genetic story seems to mirror the biblical account of Noah's family. These twin strands of paternal and maternal DNA have inspired researchers to look even deeper, examining ancient human genomes and asking whether the earliest populations might truly reflect the descendants of Noah. The possibility that genetic science could reveal traces of a story thousands of years old adds a layer of intrigue, making the next discoveries both mysterious and potentially groundbreaking. Ancient DNA interpretation from a biblical creation perspective. The study of ancient human DNA has revolutionized our understanding of humanity, revealing traces of Neanderthals, Denisovans, and other archaic humans in the genomes of modern populations. From a biblical creation perspective, researchers have begun asking a striking question: could these ancient lineages also descend from Noah and his family? In a 2024 paper published in the Answers Research Journal, Marshall Jordan explored this possibility by reanalyzing the Y chromosome data of Neanderthals and Denisovans. Instead of rooting the Y chromosome tree in a distant common ancestor shared with chimpanzees, Jordan proposed a Noah root, suggesting that all human male lineages, including those of Neanderthals and Denisovans, ultimately trace back to Noah's sons. More specifically, he hypothesized that these lineages may descend through the line of Ham, one of Noah's sons. One of the key ideas in this reinterpretation is the high mutation rate hypothesis. Traditional analyses of ancient DNA often assume that long branches in the Y chromosome tree indicate hundreds of thousands of years of divergence. Jordan, however, argues that these long branches could instead reflect faster mutation rates in certain lineages. In other words, the genetic differences between Neanderthals, Denisovans, and modern humans might accumulate more quickly than previously thought, making it possible to explain their distinct DNA patterns within a young Earth post-flood timeline. This perspective allows creationist researchers to reconcile the presence of seemingly ancient genetic variation with a biblical framework, suggesting that these lineages are not evidence of deep evolutionary time, but of rapid diversification after the flood. This reinterpretation has profound implications for understanding post-flood human populations. If Neanderthals and Denisovans were descendants of Noah, they would not represent a separate species in the evolutionary sense, but rather branches of humanity that spread and diversified rapidly after the flood. From a creationist standpoint, this perspective repositions these ancient humans within the biblical narrative, suggesting that the post-flood world was more genetically diverse and complex than often imagined. It also offers a framework for incorporating evidence from ancient DNA while still affirming a young Earth and the historical reality of Noah's family. The study encourages a rethinking of how early human populations interacted, migrated, and contributed to the genetic makeup of modern humans. It challenges the assumption that Neanderthals and Denisovans are entirely separate from Homo sapiens, and instead proposes that they may reflect lines descending from Noah's sons, carrying unique Y chromosome variations that survived through the generations. The idea that Neanderthals and Denisovans could trace back to Noah opens new avenues for research and debate. If these ancient lineages are indeed post-flood descendants, it changes how we interpret human history, the distribution of genetic traits, and the survival of unique genetic signatures over millennia. The possibility that these ancient humans preserve a direct link to Noah's family raises deep questions about the early post-flood world, the speed of population growth, and the preservation of lineages that continue to echo in our DNA today. If Neanderthals and Denisovans really trace back to Noah, what does that mean for our understanding of post-flood human history and the survival of these ancient lineages into modern times? The answers promise to reshape how we see the intersection of faith, history, and genetics, critical assessment, and implications. The idea that all modern human Y chromosomes could trace back to Noah and his sons has sparked intense discussion among both historians and scientists. From the perspective of historians, the concept of a single family origin for all humanity raises questions about the historical plausibility of such an event. Ancient records and archaeological evidence show a much more complex picture of human settlement and migration than a simple dispersal from a single point in the Near East. Historians point out that civilizations in Mesopotamia, Egypt, and other regions were already established long before a global flood, according to conventional chronologies. This suggests that even if Noah were a historical figure, the claim that his son's descendants repopulated the entire world is highly improbable. Geneticists and evolutionary biologists also approach the Noah Y chromosome hypothesis with caution. One major point of criticism is the assumption of mutation rates. Creationist studies often use higher mutation rates derived from pedigree-based analyses to compress the timescale of Y chromosome diversification to around 4,500 to 6,000 years. Mainstream scientists argue that these rates do not match broader population-level studies, which typically place the divergence of major Y chromosome haplogroups tens of thousands of years ago. Historians are also concerned about the lack of corroborating archaeological evidence. While creationist researchers point to genealogical correlations in Genesis and the patterns in modern DNA, mainstream historians note that there are no artifacts, inscriptions, or other physical records that confirm the existence of Noah's family or a global flood. The blending of theological assumptions with genetic interpretation is seen as problematic because it treats Scripture as evidence rather than as a separate narrative. Despite these criticisms, some scientists acknowledge that the studies raise interesting questions about population bottlenecks and human migration. Small populations in history can indeed leave detectable genetic signatures, and Y chromosome analysis is a legitimate tool for tracing paternal lineages. From this perspective, even if the Noah hypothesis is unlikely, it highlights how genetics can reveal patterns of ancestry and expansion over thousands of years. It encourages further research into the history of human populations and the mechanisms that drive genetic diversity. Historians question the historical plausibility and the lack of material evidence, while geneticists critique the assumptions about mutation rates and tree rooting. Yet the research has sparked broader conversations about population genetics and the ways ancient events can be reflected in modern DNA. While few mainstream experts accept the Noah Y chromosome hypothesis as factual, the studies have undeniably pushed discussions about human ancestry into provocative and sometimes controversial territory, showing the complex intersection of science, history, and faith. What do you think about the possibility of Noah's story leaving a mark in our DNA? Share your thoughts and theories in the comments below.